Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine the daily struggle faced by millions worldwide, a lot of whom are women, dealing with this relentless dryness. Dry mouth, dry eyes from Sjogrens syndrome.
Yeah, it's debilitating. Makes simple things like speaking or eating, even sleeping really tough.
And for ages, the precise reason why this happens, I mean, we knew the immune system was attacking the glands, right?
Right, the moisture-producing glands, but the actual mechanism, how it shut down the moisture at a cellular level, that was murky.
People suspected these, zipper-like things between cells were involved.
Exactly, these intercellular junctions. But what was breaking them? Could they be fixed? That was the key question.
Okay, so today we're diving deep into some pretty exciting research that seems to offer new hope. A study identifying a key mechanism and even potential treatments.
That's right. We're looking at a health news report and, crucially, the original scientific paper itself, published in the International Journal of Oral Science.
And our mission is to really unpack the core findings, right? What causes the dry mouth, and what are these potential new therapies?
Precisely. We want to get to the heart of it.
Okay, so let's get started. We know Sjogrens involves the immune system attacking salivary glands, tear ducts, causing dry mouth xerostomia, and dry eye xerophthalmia.
And the suspicion was these tiny zippers, these tight junctions between cells, were failing. But why?
And this new research from Peking University, published just recently, March 19, 2025, it has an answer.
It really does. They pinpointed a specific protein. It's called tricellulin.
Tricellulin.
Yeah, and it acts like a clasp, specifically where 3 glandular cells meet. And the key finding, this protein, this clasp, gets destroyed very early on as Sjogrens develops.
Wow, early on, like a critical fastener just popping off.
Exactly like that, a fundamental breakdown.
So how did they figure this out? What was the methodology here?
Well, it was pretty comprehensive. They took several approaches. First, they examined human tissue, salivary gland biopsies from patients diagnosed with Sjogrens using the standard 2016 criteria, and compared them to healthy controls. They looked at gene expression, protein levels, including tricellulin, of course.
Makes sense. But they didn't stop at human samples, did they?
No, they also used animal models, specifically non-obese diabetic mice.
The NOD mice. They're a standard model for Sjogrens, right?
That's right. They develop a very similar condition. So the researchers tracked changes in their salivary glands over time, 7, 14, 21 weeks.
Watching the disease progress.
Exactly. And then they did something really clever. They created genetically modified mice.
The knockout mice.
Yes, salivary gland acinar, cell-specific tricellulin conditional knockout mice. It's a mouthful, but basically.
They switched off the tricellulin gene just in the salivary gland cells.
Precisely. So they could see the direct effect of losing tricellulin in those specific moisture-producing cells.
That sounds incredibly direct. A clear way to see its role.
It really allowed them to isolate its function, but they also used cell culture.
In the lab.
Yep. Studied a line of rat salivary gland cells. This lets you control the environment and really dig into the molecular nitty-gritty.
What do they do with the cells?
They expose them to interferon gamma.
Okay, that's an inflammatory molecule, right? Known to be high in Sjogrens patients.
Exactly. Think of it as one of the signals driving the inflammation. So they wanted to see if it was linked to the tricellulin problem.
So human samples, standard mouse models, these special knockout mice, and cell cultures. Quite the lineup.
And didn't they also test some potential fixes in the mice?
They did. In the standard non-obese diabetic mice, the ones that naturally get the Sjogrens-like condition, they tested 2 things.
Which were?
One was an investigational drug, AT-1001. It's known to kind of seal up these leaky, tight junctions between cells.
Okay, like a sealant.
Sort of.
Yeah.
And the other was a molecule designed to block something called microRNA 145.
MicroRNA, that's a tiny bit of genetic material that regulates things, right?
Exactly. It can silence genes, or in this case, target proteins for breakdown. Okay, so quite a thorough investigation. What were the main results? The big takeaways?
Well, first off, they saw significantly lower levels of tricellulin pretty much across the board.
And?
In the human patient samples and in the non-obese diabetic mice compared to healthy controls.
Consistent finding them.
Very consistent. And crucially, in the mice, they saw this drop in tricellulin early in the disease process, before things got really bad.
So it's not just a late-stage consequence of damage. It might be an early driver.
That's what it suggests. It's not just inflammation floating around. There's a specific structural breakdown happening early.
Did they definitively link losing tricellulin to the dry mouth symptom itself?
Yes, that's where the knockout mice were key. The mice engineered to lack tricellulin just in their salivary glands.
What happened to them?
They developed hyposalivation, meaning reduced saliva production, clear dry mouth symptom.
Yeah.
And they also found more leakage of large molecules like albumin into the saliva.
Meaning the gland barrier was leaky.
Exactly. The junctions weren't holding tight, and under the microscope, the gaps between the acinar cells, the actual saliva producers, were visibly wider.
So no tricellulin clasp, leaky junctions, less saliva. It connects.
It really does provide that direct link.
And what about that inflammatory signal, interferon gamma, and the microRNA? How do they fit in?
Ah, that's a fascinating part of the mechanism they uncovered. It seems interferon gamma kicks things off.
The inflammation signal flare.
Right. It activates a pathway inside the cell called the JAK-STAT1 pathway.
Okay, a signaling pathway.
Think of it like a chain reaction. Interferon gamma flips the switch on JAK-STAT1, and that pathway then tells the cell to produce more of this microRNA 145.
The molecular scissors.
You got it. And microRNA 145 then specifically targets the tricellulin protein and degrades it, breaks it down.
So inflammation leads to signals, leads to scissors, leads to broken tricellulin clasps.
That's the chain of events they mapped out. And what's really compelling is when they block that JAK-STAT1 pathway in their experiments, or when they use that inhibitor for microRNA 145,
What happened?
they could prevent the drop in tricellulin levels.
Wow. So you can interrupt the chain.
Seems so. It suggests targets for therapy beyond just general anti-inflammatories.
And you mentioned they tried to reverse the damage, too, in the mice, with AT1001 and the microRNA inhibitor.
Yes, in the non-obese diabetic mice that already had the condition. And both approaches showed success.
How so?
Both the AT1001, the junction sealer, and the microRNA 145 inhibitor managed to restore tricellulin levels closer to normal.
And did that help the dry mouth?
Yes, they saw improved saliva secretion and less of that macromolecule leakage. The function actually got better.
That's really significant. It feels like this changes the picture quite a bit for Sjogren's.
I think it does. The implications are potentially huge.
Can you spell some of those out?
Well, first, just understanding the disease better. It shows that this tricellulin loss is a key early event driving the dry mouth. It's not just inflammation, it's structural damage.
A specific failure point.
Right. Which then leads to thinking about diagnostics. If losing tricellulin happens early,
Maybe you could detect that loss early, before too much damage is done.
that's the hope. Earlier detection could mean earlier intervention. And then, of course, there are the new therapeutic targets.
Right. You mentioned the JAK-STAT pathway, the microRNA.
And tricellulin itself, potentially. Or ways to bolster it,
Yeah.
like with AT1001.
And that drug, AT1001, you said it's been tested before.
Yes, it's been looked at for other conditions where gut barrier function is compromised, like celiac disease. It targets tight junctions.
So repurposing it could be faster than starting from scratch.
Potentially, yes. Because some safety data might already exist. It could fast-track things, though obviously trials specifically for Sjogren's would be essential.
And the microRNA approach.
That's exciting because microRNA therapies can be highly specific. You're essentially targeting the molecule that's doing the damage to tricellulin. Very precise.
Could this research apply beyond Sjogren's too?
It's possible. Any condition involving damaged glands or leaky epithelial barriers, maybe some forms of dry eye, certain gut issues, the role of tricellulin or similar junction proteins might be relevant there too.
Okay, this all sounds incredibly positive. But research always has limitations, right? We need to be realistic.
Absolutely. Crucially, this is primarily mouse and cell culture work.
Needs human trials.
Definitely. What works in mice doesn't always translate one-to-one in humans. That's always the big hurdle.
Okay, what else?
Well, Sjogren's is complex. It's an autoimmune disease with many factors at play. While tricellulin is clearly important, it's unlikely to be the only thing going wrong.
Right, not a magic bullet, perhaps.
Probably not on its own. And the study itself pointed this out. Remember how AT1001 and the microRNA inhibitor improved saliva, but didn't really reduce the inflammation itself in the glands?
Ah, right. So you might fix the leak, but the underlying immune attack is still happening.
Exactly. It suggests targeting tricellulin, or its breakdown pathway, is a valuable strategy, maybe in combination with anti-inflammatory treatments. It might not be the whole story alone.
Makes sense. Any other caveats?
Just that there's always more to learn. They noted some changes in other pathways, like toll-like receptors, that need more investigation. And there was a slight difference in their micro-ONA 145 findings compared to one previous study, possibly due to looking at different disease stages or types of glands. So, more research needed, as always.
Standard scientific process.
Yeah.
Still, the core finding about tricellulin seems solid. Saw a quote from the lead researcher.
Yes, Dr. Singh Kong from Peking University. It really captured the excitement.
What did they say?
Dr. Kong said, "This changes how we think about treating Sjogren's syndrome. We're moving beyond simply calming inflammation. Now we can fix the actual structural damage in the glands."
That's a powerful analogy.
It is. They continued, "It's like repairing a burst pipe instead of just drawing the floor." What's even more encouraging is that both approaches worked, which gives us real confidence in developing patient-ready therapies.
Repairing the pipe, not just mopping the floor. That really clarifies the shift in thinking.
It does. It highlights this move towards potentially repairing the underlying tissue damage in autoimmune diseases, not just managing symptoms, focusing on the structural integrity.
This whole deep dive has been really fascinating, thinking about this one tiny protein, tricellulin, having such a massive impact.
It's incredible, isn't it, the complexity inside us, but also shows how focused research unraveling these intricate mechanisms can genuinely lead to breakthroughs.
It really underscores the importance of understanding those fundamental building blocks of health and the hope that comes from that.
Absolutely. It makes you wonder, doesn't it, what other conditions, these chronic debilitating diseases, might have similar cellular zipper problems waiting to be found.
Waiting for their mechanism to be understood and maybe repaired.
Exactly. It encourages a different way of thinking about future medicine, where restoration and repair could become just as important as symptom management. A really hopeful direction.
Well, thank you for joining us on this deep dive into the latest research on dry mouth disorder and Sjogren's syndrome.
It was great to discuss it.
We really hope this has given everyone listening some valuable insights into what's going on at the cellular level and the exciting potential for new treatments on the horizon.
And if this sparked your interest, definitely check out the original paper in the International Journal of Oral Science. Following the clinical trials that will hopefully stem from this will be key.
Absolutely. Understanding the science really does empower us all. Thanks for tuning in.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.